So
Okay.
today I'll be talking to you guys about a project that I had the opportunity to work on. It's called Elucidating the Mechanism by which mutations in envelope contribute to HIV-1 drug resistance. Next. The, go to the previous slide. Okay, great. So despite the effectiveness of antiretroviral therapy, virological failure can occur in HIV-1 infected individuals, often in the absence of recognized drug-resistant mutations. And in particular, virological failure can occur in therapy containing the integrase strand transfer inhibitor dolutegravir in the absence of changes in integrase. So this observation suggests that mutations outside of integrase may confer resistance to dolutegravir. Next slide. So here we performed de novo selections for dolutegravir resistance in vitro using the clinically relevant concentrations of 1.5 nanomolar and 3 nanomolar concentrations of dolutegravir and observed resistant mutations in envelope. This includes the mutants envelope A556T and A539V. Therefore, the objective of our project is to determine the mechanism by which ONF mutations can afford dolutegravir escape. Next slide. So here we performed cell-to-cell transmission assays to help determine the mechanism of resistance of these ONF mutants. We used a GFP encoding virus, infected Jurkat cells with this virus, and incubated this GFP positive donor Jurkat cells with uninfected target Jurkat cells. And at the end of the co-culture, we measured the accumulation of GFP positive target cells. What we found is that both the ONF A556T and the A539V mutants exhibit enhanced cell-to-cell transmission compared to wild type, as observed by comparing the solid bars to the pattern bars. And in the presence of 1.5 nanomolar dolutegravir, we observe a decrease in cell-to-cell transmission with all viruses. However, the two ONF mutants are still more efficient than wild type at cell-to-cell transfer. And overall, these data demonstrates that mutations in envelope that are insensitive to dolutegravir can be efficient at cell-to-cell transmission. Next slide. So in order to determine the mechanism of resistance of these ONF mutants to dolutegravir, we also utilize a GFP reporter virus, which allows us to quantify replication kinetics as a function of viral gene expression by measuring the percent GFP positive cells over time. And in this experiment, we show that the two ONF mutants exhibit accelerated replication kinetics as compared to wild type. And with this study, we also noticed that at days of peak replication, the two ONF mutants contain a higher percentage of GFP positive cells as compared to wild type. And the graph on the right depicts the geometric mean fluorescence intensity of GFP positive cells from days of peak, and we noticed that cells infected with the two ONF mutants have a dramatically brighter GFP fluorescence as compared to the wild type. Next. And so taken together, these results suggest that in a spreading infection, the ONF mutations may decrease the sensitivity of HIV to dolutegravir by increasing the infective multiplicity of infection, known as MOI, via highly efficient cell-to-cell transmission. And you can imagine that a mechanism like this would result in more than one productive infection events per target cell. So, for example, in the absence of dolutegravir, which is shown in the left diagram, cell-free infection with wild type and mutant envelope can occur, resulting in one productive infection per target cell. However, we know that with cell-to-cell transmission, you can have more than one provirus in the target cell, and this phenotype may be more enhanced with our ONF mutants that enhance cell-to-cell transmission. And in the presence of dolutegravir, as shown in the right diagram, you can imagine that maybe the concentration of drugs that's required to inhibit cell-free infection may be insufficient to cell-to-cell transmission. And furthermore, with Furthermore, with mutant envelopes that are highly efficient at cell-to-cell transmission, the same concentration of drug might be inefficient to inhibit those mutants as compared to the wild type. Next slide. And so in conclusion, these results provide insights into the mechanisms by which mutations outside the target gene can confer antiretroviral resistance. And the study of ONF mutants that result in a decreased sensitivity to dolutegravir is of particular interest, as resistance mutations in integrase have been challenging to characterize to date. And so we speculate that these ONF mutants may provide a stepping stone on the path to high-level drug resistance in vivo. And currently, we are exploring the implications that this may have for HIV drug resistance in the context of both non-human primates as well as in patient samples. And with that, thank you for listening, and I am open to questions.
Fantastic, Phuong. I'm not sure you can put my video on now. There you go. Fantastic. Great presentation. So there are Q&As coming in the talk, but to stay on schedule, what we'll do, Phuong, is we'll invite you to answer the question directly within the Q&A tab. Does that work for you?
Yep.
I think we have time for maybe one question in case Eleanor or somebody else wants to.
Okay. Yeah, that'd be great.
Also ask an interactive one.
Yeah. So I did actually have a quick question. Thank you, Pham. So that great talk. Thank you very much. So how prevalent are these escape mutants? Just from to these antiretrovirals. I mean, is this going to be a major concern, or because there are these combination drugs in the ARVs that I wondered what if you were to speculate on.
I think that this is a great question, and I think with combinational antiretroviral therapy, the impact of the mutations won't be a huge factor, but it's something that we should look into, because the fact that mutations that are arising in envelope that are causing resistance to HIV drugs outside of the target gene is something that is a new phenomenon. And if we understand the mechanism of how these ONF mutations are able to allow these drug-resistant mutations to occur outside of the target gene, then it could also assist us in designing better drugs. But to answer your questions, we've looked at both patient samples, and there are ONF mutations that we found, but at a very low prevalence rate.
And are they restricted? I mean, presumably your patient population is, UK, or are you getting them from?
This is multiple subtypes, from subtype B to subtype C.
Okay, great. Thank you very much.
Thank you.
Thank you very much, Phuong.
Okay.